calling is currently switched to.
Hello.
Hi. So we have 1 minute more and then we will be good to go.
Okay,
I will introduce you to the Indian audiences, though many of your Indian audiences are already following you and they're a big fan of yours on Twitter.
Thank you. It's very kind of you. Let me get this set up. I'm glad to hear you're doing well, and thank you for setting this up. And I'm going to save some time for people to ask questions. In fact, hopefully, maybe most of it will be questions. Oh, shoot. Say something again. I have trouble hearing you.
So
okay.
Exactly. So we can keep it brief and then people can ask their questions because many more people will be interested, and high-ranking professors from all the teaching institutes of AIMS and PGI are there,
good.
and along with the students will be willing to understand how the clinical trials have to be read out in a lower middle income group countries to better serve their own people with a limited resources. And it's great because that's how I got hooked to your Twitter handle of arguing why this trial should be used and why should not be, why this expensive drug when it gives 4.3 months of survival, should I be utilizing or even giving hope to people? So it will be a nice presentation. So we have almost got 250 registrations from Cambodia, Nigeria, Uzbekistan, and probably some of the Iraq people, but this being an Eid, so majority of them might not be able to join today, but they have registered and they've conveyed that they'll be listening to your recorded version because it will be going on the YouTube with your permission.
Sure.
So it's almost 7:30.
Yeah.
It's 7:31. Let me introduce Vinay Prasad. He's an American hematologist, oncologist, an associate professor at Oregon Health Science University School of Medicine. He's a noted critic on direction of cancer research, and many of us actually knows him through Twitter handle, where he's voracious about how things needs to be done. He has written, and first he has co-authored a book which must be read. Probably I will say is a $30 book, which is still not available in the country, Ending Medical Reversal. Though people who have got Kindle, they can actually go on and buy that. But I've actually requested one of my cousins to send it from US. And the second book, Malignant, which I really love to read, How Bad Evidence and Bad Policies Harm People with Cancer. It's a very, very catchy word because Indian media is after either way around against the cancer doctors about escalating the cost to their benefits without realizing that whatever the drug controller gives us, we utilize this. So we'll be happy to listen to him. The more important thing is, his podcast about needs to be listened to understand how a particular trial can actually help people without putting them into a financial bankruptcy and to keep their hope without robbing them. So that's the most important plenary session. If you are a big fan of him, please go and listen to plenary session at your leisure. It will be a great insight. Over to Vinay. Virtual platform is yours and India is ready to listen to you. Thank you.
Well, thank you so much for having me. It's my pleasure to be able to talk to all of you. And that was a very kind introduction. And I guess I'll think about ways to sort of make the content more accessible in India, but I guess the best ways are probably, much of my thinking is in my Twitter feed, which is accessible everywhere. And the podcast plenary session, which is freely available on the iTunes store or anywhere podcasts are found. The book I'm gonna have to work on, think of new ways to get the book broadly available. There is an audiobook version that I recorded and it's at a lower price. And I don't control the price. I wish I could, otherwise I would just lower it all the way down. But that might be a quicker way to get access. And I read the whole audiobook and it's everything in the book is in there. So thank you so much for having me, for inviting me, for setting this up. I'm gonna talk to you about challenges and demystifying clinical trials in oncology. I think I'm gonna keep my comments brief and then let's take questions from the audience. And then if there are not enough questions, I have a little bit more I could talk about. So this is the book that came out recently, Malignant, How Bad Policy and Bad Evidence Harm People with Cancer. I think it kind of culminates a lot of my thinking in cancer medicine, which has been on the price of these drugs, which I think is horrendous and unjustified, the benefit of the drugs, which I think is modest or marginal, and the way in which the regulators appear to have, dropped the ball to some degree. And we continually have more drug approvals. People celebrate that. What we forget to ask is how many good drug approvals we have, and that might not be changing as rapidly. So in this talk that I had for you, I just really wanted to talk about two concepts from the book, two themes. There are many other themes in the book, but I think it'll give you a flavor of the book, and I think it'll be of interest to you. One, let's take a deep dive on progression-free survival. Is that something that's meaningful to patients? We see it all the time. Or is it merely measurable? And is the reason we see so much of it because it is a great boon to the industry? And then let's talk about crossover. I think crossover in cancer clinical trials is one of the most misunderstood concepts. The problem being, it's easy to think all crossover is good or all crossover is bad. That's a very naive way of looking at it. I think the reality is there's situations where you need it and you get it and you don't get it. And there's situations where you don't need it and you get it and you don't get it. And of course, some of those pairings are correct and others are problematic. So we'll talk about that. And then if there are no questions, we'll do lay of the land. So this was a patient that I saw many years ago, and a patient that very similar to a patient that you would see. This is Mr. Jones, a 75-year-old gentleman who comes to see the physician complaining of intermittent epigastric pain and a 5-pound unintentional weight loss. So he's saying he's got a stabbing pain, it comes and goes, and for the life of him, he's just been losing weight. He hasn't really been doing anything differently. And I think Mr. Jones in the United States would get a certain type of diagnostic study. And I think this is pretty common. So what do you think his doctor did? Well, in the United States, Mr. Jones is quickly going to find himself wheeled into the CT scanner. And indeed he did. And it's a good thing he did, of course, because we found something. And finding something is often hailed as the mark of success of a diagnostic study, which we can talk about. But I think that is something that, should get your attention. Merely finding disease is not always proof that you've made someone better off. But in this case, it's plausible that we might have, because in fact, the disease that we're finding is quite concerning. Many of you who are familiar with reading CT scans will know we are looking up at the patient from the feet. These are slices of the body. This is the spleen, of course. This is the pancreas with a very enlarged head of pancreas. And there are a couple of lesions in the liver that appear problematic. And we quickly sent this patient to radiology, which in the United States, they do the bulk of the biopsies. And they performed a needle biopsy of one of those liver masses. And the pathologist came back a couple of days later, and the pathologist told us it was a well-differentiated pancreatic neuroendocrine carcinoma. It was a PNET. It was a non-functioning PNET. It was non-secretory. It wasn't putting out any hormones, but it was a PNET nonetheless. And based on the spread, it was, of course, a metastatic state 4 PNET. And the year I saw this patient was 2010, so it was now a decade ago. And we do in the United States, we like to appeal to authority. And so we quickly turned to the National Comprehensive Cancer Network guidelines. And we indeed found that this patient had a flowchart entry that fit with what he had. He had local regional unresectable disease and distant metastasis. Although you could make the case that he was asymptomatic, his belly pain actually got better on its own during this diagnostic odyssey. He had a low tumor burden, fairly. And, we didn't know about the stability of his disease, but we thought it would be reasonable to observe him and follow him closely and treat him if he needed to be treated. And so we observed him. 3 months later, he comes back and he gets another CAT scan. And, I'm not the best at reading these, but to my eye, that looked broadly comparable. Another one 3 months later, again, not too much difference. Another one 3 months later, not so different. And a year later, he comes in the office and says, good news. I say, what's the good news? You're feeling better. He says, no, the good news is the Oncology Drug Advisory Committee meeting has approved everolimus for the treatment of PNET. I said, oh boy. He was telling me there's a new drug, everolimus. It's not one of those chemotherapy drugs that I talked with him before about. It is a targeted drug, and that means it's better. And the FDA, in their wisdom, has decided to approve this drug for PNET. And the vote was 10-0. It had an overwhelming show of support. And so he was very excited about this approval. And this is what it was based on. It was based on this progression-free survival Kaplan-Meier plot. Now, this shows that everolimus has a prolonged time to progression, progression-free survival duration than placebo. The median is 11 months versus 4.6 months, and the hazard ratio is 0.35. Oncologists don't always know what a hazard ratio means, but they know 0.35 is good. It is low, and lower is better. And the PFS curve, of course, has that classic stair-step pattern. The stairs of the curve are the time intervals when assertion of the endpoint is being done, the protocol-specified scans. It's a clear PFS curve. Everolimus is the winner. Look at that hazard ratio. But the same publication reported overall survival, or how long people live. And that, unfortunately, was absolutely indistinguishable between placebo and everolimus. The hazard ratio was 1. But this was something that was, easy to overlook, easy to say, well, that's immature. It's only a matter of time before that is improved. And so the NCCN guidelines, written by many people who are consulting for the pharmaceutical industry, quickly changed the recommendation. And they say they now recommend everolimus or sunitinib or cytotoxic therapy for patients with symptomatic or clinically significant tumor burden. So they added these two. Sunitinib, of course, had a paired publication in the New England Journal on that issue. And, it forced us to take another look at the patient in our office because, we had thought he was asymptomatic. We had thought he had stable disease, but was that really the case? We got another CAT scan. He said, let's just be safe. And on that CAT scan, we could look at it and we could think to ourselves, maybe it's actually gotten a little bit worse. Maybe he's having some progression. So Mr. Jones asked me a very tough question that patients sometimes ask. Should I consider everolimus? Walk me through that decision-making, doctor. I said, sure, I'd be happy to. Well, whenever I think about a cancer drug, the first question I ask is, do I have reliable evidence that this drug improves living longer, overall survival, the most important thing? And for this drug, I would have to admit, I do not have reliable evidence that it does. In fact, I do know that 12 patients while receiving the drug died in the everolimus group and only 4 patients in the placebo group, or 2%. So there's an imbalance in death while on the drug. But I don't know that that imbalance is offset by a gain in overall survival and a net gain in overall survival. I just don't know. I know the drug, the next thing I think about is quality of life. How do you feel while on the drug? And quality of life, I believe at this time was immature, but now of course we know that there is no quality of life benefit in this study. But I do know the drug had real side effects. It had increases in stomatitis and rash and diarrhea and fatigue. Now, a decade later, we've prescribed a lot of everolimus, and we know that it is not always the easiest drug to take, particularly in a 75-year-old. But at the time, we had given less of it, but we still knew that the side effects were not trivial, nothing to be discounted. But I did know it improved progression-free survival, this nebulous entity, progression-free survival. And that was what I knew. That was the upside of it. And Mr. Jones asked me, what is progression-free survival? Can you explain that to me? I really don't have a grasp of it. And so I did my best to explain it to him. I said, we scan you. I've shown you the CAT scans. You can see what the size of the tumor looks like. Imagine we were to measure that. That's the baseline measurement. And here I show you as a sphere. Progression-free survival is the time until any one of 4 things happens. 1, the patient could pass away. God forbid, we don't want that to happen. And we certainly don't want that to be the first thing that happens to somebody, but it can. And that is a progression-free survival event. The next thing is that we could find new lesions on the scans. We know about the spot in the pancreas. We know about the liver lesions. We could find lesions in different parts of the body we didn't know about. And if we do find that, that is considered progression, a new lesion on the scan. Of course, the tumor we know about could get bigger. It could get bigger. And at a certain point, it becomes progressive disease. But what is that point? And that point is when the diameter exceeds 120%. That's the cutoff. And so if you ask yourself, do patients feel fine at 119% and feel lousy at 121%? We'll have to admit that's not the case. People feel bad at different points in time. Some people may feel bad at 105%. Some people may not feel bad till 198%. People are different. The threshold of progression was not chosen because that denotes when people feel bad or when bad things are going to happen to people. We'll talk about why it was picked. So this is just a size cutoff. The other thing that could happen is you could be a very lucky person and have response, or tumors could regress. And if they regress 30% or more, again, arbitrary line in the sand, you're considered a responder. And if they grow 20% from the smallest they ever were, the nadir value, then you've had progressive disease. So, Mr. Jones, progression-free survival is one of these four things. It's the time until any one of these four things comes first, whichever comes first. And of course, some things are worse than other things, but they're treated equally in this composite time to event primary endpoint. So if we are perfectly honest, progression-free survival is something we can measure. We're very good at measuring it in oncology. It depends to a degree on the frequency with which we scan patients. The more you scan them, the more you may artifactually find trivial differences in PFS and get those to be statistically significant through bitting them into those interval scans. And we're doing some work in that space. The next thing I should tell you, which I didn't have time to go through, but different people measure it differently. We have a number of studies where you give the same scans to different people, radiologists, medical oncologists, and they will measure the tumors differently. And some of them will say this person had a response, and others will say, no, that's shy of a response. That's stable disease. Some will say this person's progressing. Others will say, no, that's stable disease. Or some have even said that's regressing. And that is really showing you that, reading a CT scan, that's not like measuring my height. It's like measuring the width of a cloud between your fingers. There's room for judgment, for interpretation. Progression-free survival is in and of itself not a direct measure of how a patient feels or function, and it is a composite time to event endpoint. In cardiology, they have major adverse cardiovascular events, or MACE, which is like the time until death, or MI, or cardiovascular death, or hospitalization. We have a composite primary endpoint. It's progression-free survival, but we don't treat it as such because we don't always break it out. So a response is 30% tumor shrinkage. And Mr. Jones notes that, and he tells me, you know what, Doc? That just sounds arbitrary. Where do those numbers come from? And I said, I got a little story for you, Mr. Jones. I'm working on this book, Malignant, so I know the story. So 1976, Charles Mortel, a very famous Mayo Clinic proceedings doctor, he convened 16 oncologists, and he brought 12 spheres, 12 marbles, if you will. And he put those marbles on a table, and he unrolled 2 thicknesses of foam rubber. And he had these 16 oncologists take 1,900 measurements. And he wanted to know, how often does the same oncologist measure the same sphere similarly? And how often do different oncologists measure the same sphere similarly? And to do that, he was clever. He picked 12 solid spheres. They ranged in diameter from, let's say, 2 to 14 centimeters. And 2 of the spheres, 2 and 2, were the same size. Is that my next slide? Yeah, I'll come to that. And then he used the foam rubber of 2 thicknesses. And these oncologists were allowed the tool, ruler or caliper, he employed in clinical practice. So what's of note here? 1, they're all men. This was a antiquated and problematic era where there were no women oncologists. 2, they're not using CT scans. They're using rulers and calipers, which were the tools of the trade for an oncologist back then. And of course, tumors 5 and 6 were the same size. Tumors 7 and 8 were the same size. So that's a really nice, clever experiment. So he could ask, how often did 2 different investigators think the same tumor was actually different? And that depends if you count a 25% difference as the threshold difference, or if you count a 50% difference as the threshold difference. And here are the percentages. And then he asked, how often did the same investigator think the same tumor was actually different? And again, it depends on the thresholds you employ. And so what Mortel realized was that a 50% bidimensional measurement was a nice cut point where different people and the same person could reliably tell tumors apart and could agree that this tumor was the same. And thus, the cutoffs that have been codified into oncology standards were not chosen because they were the best predictors of living longer, living better. On the contrary, they were chosen because they were what men in the 1970s with calipers measuring marbles through foam rubber could reliably tell apart. They were chosen for operational reasons. And of course, my patient, Mr. Jones, is very smart. He said, wait a second, you said the response cutoff was 30%, but now you're saying Mortel used 50%. I said, well, it turned out that he did, but that was a WHO bidimensional cutoff. And if you look at the RECIST cutoff, as I show here, it's 30%. And roughly volumetrically, those are the same change. And so good point goes to Mr. Jones for catching that. But that's where we have the cutoffs. That's why we have these cutoffs to this date. They are chosen for operational reasons. In an era before modern scans, what could we reliably tell apart? And if you really think about it, is a 30% reduction, is that what we should be striving for? It should be 50, 60, 80%, 90%. I think we can talk about that. And what actually correlates with survival? So the next thing I would say is, Mr. Jones tells me that no wonder progression-free survival doesn't capture how I feel. But surely drugs that improve PFS on average extend lives. So we've done a great deal of work now over the last 5, 6, 7 years to probe that question. And we've looked at umbrella analyses of meta-analyses. And here's really what we find. If you wanted to know whether or not progression-free survival or MRD or PathCR predicts overall survival, is it a surrogate endpoint? How do you study that question? It's really important to study it the right way. And you need to do a trial level analysis. What do I mean by that? You go into a space and you collect all of the trials in that space. So every trial where they measured overall survival and MRD, every trial where they measured PFS and OS, and you collect them here. Let's imagine there's hypothetically 2 trials. For each trial, you make each trial one data point where the change in the PFS between the 2 arms is the X, and the change in the overall survival between the 2 arms of the trial, the experimental control arm, is the Y. And you scatter plot all the trials you have on a graph, and you perform linear regression. And the closer the points congregate to the line, the higher the correlation coefficient, the better the correlation. Literally what you're asking here is what % of the variability in overall survival is explained by variability in the surrogate in clinical trials that test experimental agents? That's exactly the question of surrogacy, and that's exactly what you're interrogating in this study. And this is just an example where they correlate well, and these are cutoffs that are proposed by the German group IQWiG, the quality group. So we have done this for everything. We've looked at every published study ever done in every tumor type for every surrogate, and we have created this umbrella review that we published a couple times now, JAMA Internal Medicine and the European Journal of Cancer. And this is a visual representation of that review. The diamonds are surrogate validation studies. The bigger they are, the more data points they have. The biggest have over 100, and the smallest have, I think, 4. 4 is actually not a terrific number of data points to run regression through. 2 data points, and you're gonna have a good correlation. I promise you that. I'm not an expert on regression, but that much I know. 4 is not terrific. 100, now you're talking. The color tells you how strong the correlation is, how well PFS predicts OS. Green is good, red is bad. And the timeline tells you the years in which they ran this study. What you see, of course, and this is actually truncated, there are many, many studies run in the last 5 years. The enthusiasm for finding new surrogates is rampant. It's running rampant in the field. But we also see there's just a whole bunch of red. And in fact, if you look at it another way, in the metastatic setting, response rate and progression-free survival generally do not predict overall survival in tumor types in the metastatic setting. In the adjuvant setting, it's a lot better. We could talk about that in the questions. Okay, I'm going to skip that part. Of course, after all this discussion, unpacking PFS, unpacking the lack of survival benefit, there's no quality of life benefit, what did Mr. Jones decide to do? Mr. Jones decides to do what many people in the United States decide to do, which is, let's give it a try. You know, this is the most seductive, I think, idea about the industry, which is that you can put very marginal, modest, mediocre products on the market, things like selinexor in myeloma, things like panobinostat in myeloma, some really, really questionable products. And no matter how much the grade 5 AEs, no matter how fleeting the benefit, there will be people willing to give it a try. And the entire reason why we have a regulator, why don't we just have a free market where people choose what they want, is that we believe as a society that a sick, vulnerable person with cancer is not in the best position to choose the risks of the most risky toxic drug products. And once upon a time, we allowed it, and people took all sorts of unproven remedies. And so we use the public regulator as a protective mechanism to shield people from choices that are not in their best interest. But that regulator has increasingly lowered the bar, allowing people to take choices that really are maybe not much different than choices they would have in 1910. And that to me is a tragedy. And of course, inevitably, like Mr. Jones, people want to give it a try, because if you have cancer, of course, the possibility of benefit looms large, particularly in the world of advertisements. Of course, 1 month later, Mr. Jones, like many patients, discontinues the drug, saying, he used a word I wasn't familiar with, but he felt worse. Okay, so I'm going to skip this part because I want to get to questions. Let's just talk about crossover. You know, in cancer medicine, we have a design feature in randomized controlled trials that is immensely unique, and that is crossover. And by crossover, we don't mean I take sugar pill, I take drug, I cross back over, I cross over again. No, we mean unidirectional crossover from people initially assigned to the control arm to the intervention arm. So let me show you that graphically here. Among patients assigned with cancer, they're randomized to the drug or placebo. If you progress on placebo, you get the experimental drug. If you progress on the drug, you get standard of care. So this is what it looks like. You get randomized to the drug or placebo. When you progress on placebo, you get the drug. So you're randomized to PNET patients randomized to everolimus or sugar pill. If they progress on sugar pill, they get everolimus. If they progress on everolimus, they probably get either a streptozocin or a carbotoposide kind of chemotherapy regimen. I think what we forget that crossover is desirable in some situations, but crossover is also undesirable in others. And we get crossover, or we don't get it. And thus we get this grid pattern that if we wanted crossover and we got it, well, that's a good trial. If we didn't want crossover and we didn't get it, that's a good trial. But the opposites are bad. And we get a lot of the opposites in this business. Everolimus has crossover in that study, the RADIANT-3 study. Was that good or bad? I'll use a different example. Some of you will be familiar with Sipuleucel T. It is a cancer therapeutic vaccine. It is a vaccine that we make in people who have cancer and we administer to people with cancer. And the goal of the vaccine is to elicit an immune response in the patient so your own body fights your own cancer. That's the goal of the vaccine. And in this randomized trial that led to drug approval, there was an overall survival benefit. So why could I be a critic about that? The overall survival benefit is shown here. It's, I think, 20 to 24 months, 4-month median overall survival benefit. It looks rather modest, but compared to a lot of cancer drugs, it's not the worst I've seen. But interestingly, Sipuleucel T had no responses, 0% response rate, and progression-free survival was not improved. So no responses, no progression-free survival. It doesn't appear that you're changing the dynamics of tumor shrinkage or growth, and yet survival is better. How is that possible? How does that work? And the other interesting thing about it is the only cancer therapeutic vaccine in the history of man to ever be approved. Nothing came before it, nothing came since. No response, no change in time to progression, no activity, but that 4-month OS gain. How do you reconcile this? So we got to take a close look at this study. So in this randomized trial of patients with castrate-sensitive metastatic prostate cancer, when they progressed, if they were assigned Sipuleucel T, they got docetaxel. Turns out docetaxel, since the TANAC paper, has a shown survival benefit. If they got placebo, they got frozen Sipuleucel T. If they progressed yet again, then they got docetaxel. So now you have something that's interesting here. You have an imbalance in docetaxel. Docetaxel is life-saving. If you got assigned the study drug, you get it sooner than if you get assigned placebo. You get it later. So you've imbalanced post-protocol therapy, and one in particular that improves survival. And in fact, that imbalance is is proven in the publication because only 71% of the placebo, only 57% of the Sipuleucel T group, let me put that another way. 57% of people who got Provenge, the vaccine, they got docetaxel, but only 50% of the placebo group. In the Provenge group, it was 12 months, and in the other arm was 14 months. So basically, it looks like this. 57% got it here, 50% got it here. And it took 12 months here, and it took 14 months here. So this is a problem, because now the question is, does this arm live longer because of the Sipuleucel T, or does this arm live longer because 7% more got the drug, got a life-saving drug, and they got it sooner? And you can't tell those two hypotheses apart. And that's what the AHRQ says. They say, we cannot exclude the fact that survival in the absence of response rate or PFS is actually due to harm to the control group from a delay in chemotherapy due to getting an ineffective frozen salvage product. So what's the take-home lesson? The take-home lesson is crossover is undesirable in trials that test the fundamental efficacy of drugs, such as vandetanib in medullary thyroid cancer, such as Sipuleucel T, such as everolimus in PNET. It is undesirable. You don't want to cross over people because you can confound overall survival. In the case of Sipuleucel T, you may have created a spurious survival benefit that is not actually driven by the drug itself, but just by an imbalance in good post-protocol therapy. You don't want crossover in these situations. And when you get it, it really muddies the waters. Let's go to the other example where you wanted crossover and you didn't get it. So this is the same setting, metastatic castrate-sensitive prostate cancer. Abiraterone versus placebo, the latitude trial. There's a clear benefit here. Abiraterone improves overall survival over placebo, but we forget that prior to this study, we already had the Cougar trials, and we were using abiraterone both pre and post docetaxel in the metastatic castrate-resistant setting. And thus, Johan de Bono and colleagues write this letter in the New England Journal of Medicine. Before these trials were run, the standard of care for patients with advanced prostate cancer included sequential androgen suppression with various life-prolonging therapies, such as abiraterone and enzalutamide. However, the control regimens in these trials were not designed to include the current sequential standard of care. These treatments were not specified in the protocols. This is critical since the majority of men in the control arms died without exposure to abiraterone or enzalutamide. Thus, the drugs used in these control groups were inconsistent with the current prevailing standards of care. This has implications for the conclusions of the trials and raises questions regarding whether or not there was a benefit for all participants. So let me put that to you another way. What they're saying is, you had a drug that you know works in an advanced disease setting. In fact, it has an all-cause mortality benefit. And you want to know if it works better upfront than the advanced disease setting. So you randomized upfront versus standard of care. That's reasonable. But in your standard of care arm, you didn't give the drug in the advanced disease setting where you're supposed to give the drug, where it is standard of care. You didn't give it at all. So there is a survival benefit, but I don't know if that means I should move your drug early or if I could still have gotten the same survival by giving the drug later. I don't know because you didn't give it. So crossover is desirable when a drug already has proven benefit in latter lines of therapy and the trial is testing moving it to the front line. And yet we routinely see that that does not occur. We see that in a number of clinical trials where, in fact, I bet we're going to see it in the ADAURA study. Everyone is celebrating ADAURA for that DFS benefit, and I suspect it will have an OS benefit. But if you are in the control arm of ADAURA, you should be getting the standard of care in the US, which is osimertinib in the frontline setting there. Why? Because if you can afford osimertinib in everybody, you certainly can afford osimertinib for the people who progress in the control arm. If you can't afford osimertinib, which is another problem, then you shouldn't be even trying to attempt it in the adjuvant setting. But I suspect that trial is going to have a lot of gefitinib and erlotinib given in the frontline setting. And that is not the question that we really want to answer. We want to know if the routine upfront administration is better than subsequent use. Okay, so this is the way I think about crossover. I'm going to stop right there. I'm going to take questions. You guys can start typing it in the chat. I see there's one already. And then, you know, we can come and talk about more stuff. But I guess I would say that, you know, what do I hope to have conveyed to you is that it is so easy to be seduced by waterfall plots. They exaggerate response rate. Response rate, progression-free survival, those are not metrics of people living longer or living better. All the new drugs improve, or not all, Siblucil T, but most of the new drugs, 2/3 of them, in fact, only improve response rate and PFS. We should ask for more for our patients. The price of these drugs we didn't get into, it's horrendous, it's unjustified. If you had that much money, there are often better things you can do with that money in resource-limited settings. Crossover is often misunderstood because people don't draw the distinction between when you want it and when you don't want it, and I try to tease that apart. And if you like the content, I think that the easiest way to access the stuff is the podcast, and then you can go from there. The introduction of the book is a podcast episode. Let me take some questions. A great topic presentation. Look forward to connecting. Thank you so much.
I can actually read for you. Censoring in everolimus arm was higher and earlier, maybe less fit of patients having more toxicity stopped the drug and were LFU. These may hike PFS in favor of everolimus.
Yes, let me explain that to people. So people really, that's a very smart point. So what is this person saying? You do a randomized control trial and you randomize people to drug or placebo. Okay. When you measure overall survival, in every single person at every point in time, one of three things has happened usually. One, they're dead. So then they've had the event of interest. Two, they're alive and they're continuing to take the drug, or they're alive and they're not taking the drug. I mean, they could be post-protocol, but they're alive and they're alive at that time. The other thing that could happen is they only enrolled on the study 2 months ago, and I'm building my Kaplan-Meier curve at 6 months. And so I have to censor them because, I don't know what's going to happen to that person 4 months from now. They only enrolled 2 months ago and now it's 6 months. So that's a reason people are censored. And at overall survival, those are the typical reason for censoring is a late enrollment. With progression-free survival, one of the reasons people are censored is if they are no longer participating in the study and getting the scans. And that's a unique reason to censor someone. That can be a reason that occurs if somebody takes the pill, feels lousy, and discontinues. And this person pointed out that in the everolimus peanut arm, just as in everolimus and Bolero, which is the breast cancer study, just as with the PI3 kinase inhibitor in breast cancer study, in many studies, the people who are getting the experimental drug, there's way more of them censored early than people in the control arm being censored. And that censoring is not from late enrollment, because if it was late enrollment, it would be balanced in both arms because they're randomly assigned at the point of enrollment. That has to be from toxicity leading to discontinuation or discontinuing the protocol. When you have more people in one arm discontinuing the protocol early, you're excluding people from the PFS calculation. And then the next part is, who are those people? Are those average people, which is the assumption of the Kaplan-Meier method, or are they people who are sicker on average? And it's very plausible they're sicker on average because that's why they can't tolerate the medicine. And so what you might have in these situations is a spurious PFS benefit driven by censoring the sickest people because the toxicity of the drug knocked out the sickest people. And that's what this person is pointing out that could have been happening in the Radiant trial, and it could have happened in Bolero, and it could have happened with this new PI3 kinase drug in the Solar 1 trial.
So another question, Dr. Abhinil Mittal has asked, sir, do you have used any drug based on a PFS benefit in advanced disease, as many drugs are approved just on that benefit in advanced disease? Because Vinay, the most important thing is there's a plethora of medications and tsunami of newer medications which are coming by every ASCO, every ESMO, and every ASH. How does one sitting in a LMIC decides upon which one to use? Because 4.3 months progression-free survival at say $20,000 cost, giving a hope that if I had money, I will have survived. In India, it is perceived as if I had money, I would have survived. So how do you take that?
Yeah,
Isn't that hyping the hopes for the people?
Yeah, I mean, I guess I 100% agree with what you're saying. That I mean, I think in a resource in a low and middle income country, in an LMIC, a drug that merely improves progression-free survival without improving overall survival. And let's be honest, the PFS benefit is not typically 45 months, not 55 months. It's typically, as you say, 4 months. Actually, in fact, I know the typical answer. It's 2.5 months because we've done, FOHO has done that study. A 2.5 month PFS benefit when someone has to pay out of pocket $20,000 US, that's not justifiable. I mean, you can tell the patient that, if you're going to have to take out a loan, if you're going to have to sell your house, if you're going to have to deny your child inheritance, I don't know if in good conscience we can recommend such drugs. People I know in these countries, they often do things like that. They sell jewelry, they sell, inheritance to pay for these medications. They feel guilty if they don't. That's not a way to, that's no way to have to go on. I mean, I think I would say very frankly, we know for certain this, we don't know this drug lengthens how long you live. We know it's not, we know it doesn't cure you. We could even have the discussion for, drugs with a proven survival benefit, but that benefit is modest. People should be allowed to make these choices. But we know the drug doesn't cure you. It doesn't have a proven survival benefit. It only has a PFS benefit. I couldn't in good conscience watch you spend your hard-earned money, hard fought for money in this way without advising you that I'm not sure it's the best thing for you. I mean, I think that's a reasonable thing in a low and middle income country to say. In the US, we have a different mentality. We have insurers pay for things like that. We tend to allow people to make those choices. But you know what? There are a lot of people in the US as well who, even if the drug is free, they say to me after I explain what PFS is and what we know and what we don't know, they say, you know what? I don't want to take that chance. And for most solid tumors, we do have some backbone, older standard chemotherapy drugs that do have a benefit. It's not that you're giving nothing, you're giving something and it does have a benefit. You're just not adding on some novel molecular drug that we have a question mark next to. So
another question, what about hype created for immunotherapy? I think small benefit for too high cost. I mean, isn't that a truth that therapeutic drugs are swaying the market towards a smaller PFS and rather than giving anything? So we talk about nivolumab and pembro being utilized now left, right, and center as off the cup thing for every single disease, whether it be head and neck, whether it be melanoma. I mean, melanoma is another different disease.
That's
but
what I was going to
anything
say.
to
Yeah.
talk about, I mean, RCC, hepatocellular, but what is the end point? Because these trials, when they come out from ASCO and JCO, and when the Pembro and nivolumab is available in the country where there's no checkpoints and people are paying out of the pocket, they feel that if I have the money, I can buy all these drugs. So what do you think about actually immunotherapeutic drugs making a dent into cancer survival?
Yeah, no, that's a great question. I mean, I guess I would say that, let me put my low and middle income country hat on, my LMIC hat. I would say that, who are the people that you need to be thinking about checkpoint inhibitors? I guess what's coming to my mind, of course, melanoma, metastatic melanoma. You're going to have a durable fraction. It's not going to be everybody, but some fraction is going to be appears to be quite durable. The other place I might think about it is maybe second line in MSI high colon cancer, because that appears to have some durability. But if you really were in a tough situation, is that the kind of thing that you need to be giving in every head and neck cancer patient, lung cancer patient, renal cancer patient, where the benefits are, there is a benefit, but is that benefit worth the price if people have to, really have financial hardship as a result? I think those are not as compelling as the most compelling indications, melanoma and I think MSI high colon. I think you're right in the sense that there's a huge hype around it. The drugs are going to be netting 20, 30 billion a year. They've already earned their money, manyfold. They certainly do benefit people. I mean, I don't want to discount that. I think probably 15% of all cancer patients will benefit from a checkpoint inhibitor. I think that's probably the fraction of people who benefit from checkpoint inhibitor. But that's not the same as the fraction that really has a long durable benefit, which I think will be less than that. And when you were talking about the tremendous cost and how much people have to suffer to get access to it, I think it's a very challenging situation, as you know. And so if you've got somebody in your clinic, lung cancer patient, low PD-L1, or head and neck cancer patient, low PD-L1, I really think that it's not going to be something that I'm going to have to, it's not going to be the top of my mind thinking about that.
So Dr. Baghel comes with this, when or where do you think PFS is a good endpoint, as OS may take time to mature?
Yeah, that's something that the industry says, that OS takes time to mature. I guess I would say that, when you're in cancer medicine and death is occurring rapidly, so one of the things they said recently was the POLO trial, POLO pancreas cancer. The OS benefit is gonna mature, but we've already reached median OS, and we know a lot about the OS, and there's no OS benefit there. I guess I would say, in the metastatic solid tumor space, in a low and middle income country setting, I think I would probably not be very eager to act upon PFS alone. I would probably want to wait for OS. I think the places that you might think about a surrogate endpoint is if you are in the adjuvant setting and there's a disease-free survival benefit for something. But there too, I think you got to be cautious, really look at the details of the trial. And I'm having a hard time with that because, in the US, the way we would handle that is very different than in a setting where these drugs are so expensive. I guess I would say that, really transformative drugs often improve both at the outset. And this idea that it takes longer for OS, we've probed that in a paper called about how long study time length and the endpoint, which came out in JAMA Journal of Medicine. So I guess I'm going to think about that question more, but I think generally I would favor sort of, you can kind of keep an eye on things when they just reach the PFS endpoint in low and middle income country settings. Yeah.
So another question, sir, what is your take on PRP inhibitors, huge PFS benefit, but OS benefit is still doubtful, even in second and third line settings, specifically in ovary and now in breast?
Yes, exactly. That's exactly my take, which is for drugs that are so miraculous and transformative, the very meager OS benefit is incredibly noteworthy. And now PARP inhibitors, of course, they're trying to put them everywhere. They're trying to put them in prostate cancer, where I think the data is lousy, and I cover that in my podcast. They're trying to put them in pancreas cancer, where they give you 4 months of FOLFIRINOX and stop it, and then randomize you to olaparib or placebo and have a PFS benefit. And even in ovarian cancer, I think. So, if you really feel compelled to use that, I think taking into account HDR status and BRCA status might be a more prudent thing to do it. Whether or not everybody needs it in the maintenance setting, the front line, I think is a question mark, but maybe to reserve it for latter line of therapy. But that person is right that the OS benefits have not been commensurate. You know, there we're talking often PFS gains of 20 months, and look at the OS benefit. It seems to be quite on the fence or a marginal benefit. I think now there've been some positive results, but you know, they're not the whopping OS benefits that we were hoping for.
So what, according to you, is a better surrogate endpoint than PFS? Time to subsequent therapy in advanced disease or EFS in adjuvant setting? What is your take on surrogate endpoints other than PFS?
Yeah, I think that person will really like to read the book, because the answer is going to sound jarring to you, but hopefully after 200 pages of me laying out the arguments, you will see the answer is much more parsimonious. The answer, I really think, is that we need to be measuring survival more often. If we measure survival more often as the de facto regulatory endpoint, everything will shift. You know, pertuzumab in Cleopatra, the trial was run pertuzumab, trastuzumab, taxane versus trastuzumab, taxane. It was run in the frontline metastatic setting. That's the first trial for pertuzumab's approval. And it was approved based on PFS. And they say, well, if we had waited for OS, it didn't mature for another year and a half. It would have taken so long. And I say, if you had really asked your company and said, we're going to ask you to prove OS benefit, they're not going to go in the frontline metastatic breast population. They're going to go into the third line population, show benefit. And then when they want to move it to second line, think about my crossover part of my talk. They're going to have to prove that routine second line administration is better than third line administration. That's an uphill battle. So I think that, you know, in terms of like, how do you solve drug regulation? The more you ask for people to improve the endpoints that actually matter, you will change drug incentives in a massive way. You're not going to pursue compounds with no single agent activity. You're going to pursue compounds in latter lines of therapy first, then try to march them up. You're going to change everything. And so the problem with these surrogates, from MRD to EFS to PFS, is just that they have a huge rate at which they don't correlate with survival. And what that means is we can approve 60 drugs a year, but maybe only 10 of those drugs actually improve survival and quality of life, and the other 50 of them just take money from patients' wallets. And there are maybe other things we could be doing with that money, such as providing better nursing care, better social support, things like that. And so I guess I think that the answer is not the answer is not the perfect surrogate. It's sort of more rational drug approval. Anyway, in the book, I try to make that case at length, and by chapter 14, I hope to have made it.
Like cancer like myeloma, where CR is the best CR as well as quality of life is concerned, prolonging duration of CR is more important. Don't you think prolonging PFS, giving the most potent drugs upfront, is more important than OS? And actually speaking, in my clinic, every time I tell, I choose a triplet, telling myeloma that, okay, walk through a journey till 2023, and then you might have a miraculous drug which might be coming. Because we know the CAR T-cell is not there. So the best PFS has to be chosen. Might be expensive now, but if I can give you a 60-month PFS, like a transplant, because there's a huge debate which goes on between VRD and a transplant.
Yeah.
So giving a larger progression-free survival might entail that the economy might become a 5 trillion one, and we might have enough money to get CAR T-cell at the current cost.
Yeah. That's well put. I mean, so I think both of these are common arguments, and let me try to summarize what I think the argument is. Let me summarize your argument first, which is even if a drug doesn't improve survival, if a drug improves progression-free survival substantively, it may serve as a bridge to a new drug hitherto undiscovered, and that's not yet out yet. A new drug may come, and by giving a long PFS, I give my patient a better opportunity to benefit from new transformative drug. That's an argument. And I guess I would say, I mean, in my thinking of that argument, I guess I would say, if there is a new transformative drug, be that BCMA CAR T, which to be honest with you, from everything I've heard, BCMA CAR T has got a 100% relapse rate within 10 months. You know, I'm hearing it's not durable, but anyway, but let's just say there's some new drug that's going to come that's going to be really good. What is the prerequisite to benefiting from that new drug? What is that prerequisite? Is it having your disease progress later or not having progressed yet? I don't think that's the prerequisite. The prerequisite is being alive and having a good functional status. That's the prerequisite. So you could imagine, you could have kept the patient going. They could have progressed 3 times, got, Revlimid, dexamethasone, they could have got carfilzomib, dexamethasone, maybe they got, maybe they got Dara Rev Dex at the end. And as long as they're alive and they don't have, end organ dysfunction, I think they're a candidate for that compound. So you could have given potentially two scenarios. One, you've strung them along with RD, carfilzomib dex or Velcade dex, and then Daradex. And then they get the new drug. Or you gave him Dara, carfilzomib, Revlimid, dexamethasone upfront 4-drug combo, like some of these myeloma doctors do, and then they still haven't progressed and they benefit. So I guess what I think is, I just think it is sort of a fallacy to believe that drugs that improve PFS mean you're more likely to get a Deus ex machina, a magical drug in the future. I think all you have to do is be alive and not have end organ damage. So I'm not so sure about that argument. The next part of the argument is, what about quality of life? I guess myeloma is a great space, to talk about this, because there's no space that, in my opinion, has been more hijacked by the industry than myeloma. The rules for advancing drugs in myeloma are very different than other tumor types. They really, and it's reflected in a few things. The people in myeloma who are the thought leaders, with the exception of, I think, people I can count in one hand, they're among the most heavily conflicted doctors for the industry. You can look them up on, open payments and see how much money they're getting from these companies. They're all getting money from every single company. And their mantra is always the same: more drugs earlier, sooner. If in the future, they're going to give MGUS patients Dara RD, and it'll be the time until myeloma. Or even better, they give everybody Dara RD, and it'll be the time until MGUS. That's the end point of the trial. They really like giving more drugs sooner indefinitely. That's the mantra. And there are a number of elaborate reasons why that is beneficial. And one is that even if we don't improve survival, we improve the quality of life. And what I would say is that there is a way to test that hypothesis, and that is to routinely administer health-related quality of life questionnaires in these studies for a long period of time. And I don't see an enthusiasm to actually do that. So I guess I would say in myeloma, this is a fallacy of surrogates, because PathCR is better than no PathCR. But drugs that improve PathCR don't always make you live longer or have better DFS. So it's called a surrogate paradox. Getting into a CR, of course, is better than not getting into a CR. But drugs that marginally improve CR rates may not make people live longer, because the people who weren't going to be in CR, who you flip into CR, they may have the disease come back a little bit faster than average because they have bad biology, and that may negate any benefit you think you're getting from driving them to CR. And of course, everything we have in myeloma, CR, MRD, every measure, there is no measure that measures the last cell in the body. These are all different imperfect ways to measure myeloma in the body. Even when you flow, 4 times 10 to the power of, 7 or 8, you flow tens of millions of cells looking for myeloma, 1 cell in 7 million. You still don't know beyond that level of detection. Okay, so anyway, so this argument in cancers like myeloma where CR1 is best, to my mind, in a low and middle income country setting, I guess in the US, I tend to favor, triplet, I think, is the standard of care. VRD is the standard of care. But if you really had a gun to my head and you had an older, frailer person in my clinic, you had an older person, do I really know for sure that Revlimid, dexamethasone with close follow-up and then giving them all the drugs in sequence, that's really inferior? I guess I don't really know that for sure. And so if cost is an issue, I would be okay with doing that, I think. So that's just my thinking on that topic. I don't know. I think it's something we could debate for a whole hour.
So before we take another 10 minutes, we have to go. Another 3 questions. Dr. Pankaj and Dr. Dinesh Burani and Dr. Tulika said, any comment on this myeloma space Dr. Vinay has just raked up in terms of saying, okay, RD, VRD, surrogate markers of CR, PR? Anything which you'd like to say or comment upon it?
I guess one thing I would say is look at how enthusiastic they were that carfilzomib, Revlimid, dexamethasone was going to beat VRD. For years in the US, we already switched to carfilzomib because everyone said it's better. And now look, we have the new study came out at ASCO this year and it's not any better. That just goes to show you that I think that people oversell these drugs. Okay, the next thing I would say is look at ELO RevDex. ELO has a small survival benefit in like the 3rd line study, but it failed in 2 frontline studies. So maybe it's not as terrific as people think. The drugs I like in the space are Dara, of course. It has a strong response rate, CD38 antibody. I like Velcade, Revlimid, dexamethasone. I think, I think, to be honest with you, when you talk about transplant in CR1, my thinking is that in the age of all these modern drugs, that we don't necessarily know that transplant improves overall survival. That's all in the prior era. And so I think I am a little bit more in mental equipoise about that decision. And so if it's in a setting where transplant has a high transplant-related mortality, or it's hard to get, or it's expensive, I don't think I would push for it so hard. So I guess my thinking is that, being very attentive to somebody, giving them VRD when they progress, giving them DARA-RD or DARA-POMDEX or something like that. I mean, I think you can string those triplets together and get, very good outcomes in low and middle income countries. One of the challenges is that there's a temptation to always chase what the thought leaders in the US are doing. But I would sometimes say that just because they're thought leaders in the US doesn't mean that they are always right, whatever the cancer is. And so countries like India have done some really amazing studies, and we shouldn't discount that. And physicians in India, I think, are superb, incredibly thoughtful, and they have a lot of experience. And sometimes if you really were to put the chips down, you might have much more experience than some of these quote unquote thought leaders. So I guess so those are just some general thoughts.
Can I talk?
Yes.
Yeah, please, Dr. Dinesh.
Yeah, I was just saying the DARA
Dr.
is
Dinesh.
coming in. Yeah,
Dr. Dinesh,
yeah.
just a
Hi,
minute.
I'm Dinesh Burani from Rajiv Gandhi Cancer Institute.
So Dr. Dinesh Burani is one of the senior most physicians.
Let me talk. Let me
of
talk.
one of the largest cancer
Let me
hospital
talk.
in North India. That
Dr.
I
Rahul
have to
Bhargava.
tell.
Okay.
Please, Dinesh.
The important is now that probably the DARA will be coming up front. So DARA is hugely expensive. Considering the VRD has a very good results, and putting the DARA in the first relapse probably will be more useful in our country rather than giving the DARA out to everyone.
Of course.
Maybe. So I think I'll be worried, and I'll agree, just because of one trial, or probably that trial is on the way, the phase 3 trial.
Yes,
and the DARA will be everywhere, and
Yes.
everyone probably in India will be putting everyone into the financial crisis, rather than allowing them to live longer.
Yes. So I just want to emphasize what you're saying, which is very wise. In the US, we're doing a frontline study, VRD plus or minus DARA, and I have zero doubt the PFS is going to be a win. I'm pretty confident Janssen's going to get a big win out of this. And in the US and Western Europe, we're going to start giving DARA VRD. But this person's point is, you have not yet proven that giving DARA VRD is better than what the control arm should be, which is VRD and then DARA at progression in second line. And in fact, I wouldn't be surprised, I don't know this study, but this is an example I talk about, crossover. You want crossover because you want to ask, is routine upfront administration better than sequential? But I won't be surprised if that study I showed you, that they don't have great second line use in this trial, that they do have an OS benefit, and it's going to be widely kind of misinterpreted, I think. So I think Dinesh is right. The right way to think about this is, even if that study shows a PFS benefit, doesn't necessarily you need to do it in a low and middle income country. The real question is giving all 4 drugs up front better than giving them sequentially? And in terms of overall survival or health-related quality of life across the time horizon of multiple lines of therapy. And I don't think that they're going to interrogate that question in an adequate way. In fact, I'm confident that they won't. So that's a very wise comment. People should heed that in general, and this is true of everything in cancer space, that just because there's some new sexy trial that shows giving more drugs upfront is better, you have to ask yourself, did they really prove that it improves longevity or quality of life over the patient's entire cancer journey? And is it worth the price? That's the real question. So thank you for that.
So Dr. Tolika, Dr. Pankaj, Dr. Tapan Saikia.
Okay, I can make some comment. I am Pankaj Malhotra from PGH Chandigarh. So what I believe is that, as you become senior, you start realizing that, the trials are only a guide to you. When you are taking clinical decision in a given patient, then you can keep all the trials in front of you, but then you have to, see the pros and cons of each trial and then take a decision in a given patient. Yes. I don't think so. You just, this trial is showing this, so you just apply that trial on the patient. So yes, as a doctor, you have to, make a call. And as you have already mentioned in your slide about the PFS and crossover, all those things. So we should make the physicians more knowledgeable about these trials, and then take a relevant decision in the clinic. Thank you.
Thank you so much. My whole career interest is exactly what you say. which is that we are not automatons mindlessly carrying out the results of clinical trials. Although some of us are, that's the problem, but we should not be. We should aspire to be people who independently, as Dinesh pointed out, how do you think about the study? Does it apply? Does it make sense where I am with the resources my patients have? Does it make sense? Is the benefit really worth the price, as Rahul has continued to point out? You know, we have to ask ourselves these tough questions. And, you know, to be honest, you know, that's the point I wanted to make. Indian physicians are often, you know, very excellent. You're thoughtful people. You know the realities of the situation, and sometimes it's not always what the key opinion leader says to do. It's not always what's best in your country, in your situation. So thank you for reinforcing that. I think we have to have healthy skepticism, and I hope in my podcast and in the book to teach some of the other ways in which we are actively trying to be manipulated, I think, by clinical trials.